Good afternoon, everybody. My special thanks goes to the organizers for giving me the opportunity to present some of my recent work. One major focus of my lab is to study the metabolic roles of G-protein coupled receptors, or short GPCRs, in modulating different metabolic functions under physiological and pathophysiological conditions. Today, I will talk about the regulation of glucagon release by different GPCR signaling pathways. The ultimate goal of this work is to identify new targets for the treatment of type 2 diabetes and related metabolic disorders. To do this work, we generated and phenotyped a very large number of novel mutant mouse models. As you all know, GPCRs represent a superfamily of receptors that are activated by a large variety of extracellular ligands. As shown here, when activated by an extracellular agonist, GPCRs are able to activate different functional classes of heterotrimeric G proteins, which are subclassified into 4 classes: GS, GI, GQ, G12. The G proteins are named after the alpha subunit contained within the G protein heterotrimer. The activated alpha subunits are then able to modulate specific intracellular signal transduction pathway and ion channels, ultimately leading to the desired cellular response. Like all other cells, metabolically relevant cell types express dozens of different GPCRs. However, the individual GPCRs are also expressed by many other cell types and tissues. Moreover, selective ligands with high selectivity for a particular GPCR are frequently not available. For this reason, it has been extremely difficult to explore the in vivo roles of a specific GPCR or a specific GPCR signaling pathway in a particular cell type. To overcome these difficulties, we started to use a group of designer GPCRs known as TREADs as novel tools for this kind of research. The term TREAD has been coined by Brian Roth at the University of North Carolina quite a while ago. It stands for designer receptor exclusively activated by a designer drug. Chemically, structurally, the most commonly used TREADs are mutant muscarinic receptors, which, due to a pair of point mutations within the transmembrane core, are no longer responsive to acetylcholine, which is the endogenous agonist on muscarinic receptors. However, these TREADs can be selectively activated by a synthetic drug called clozapine N oxide, or short CNO, which activates these receptors with high potency and high efficacy. Most importantly, CNO is pharmacologically inert, at least when used in the proper concentration or dose range. We are now fortunate to have all 4 TREADs that couple to each of the different major classes of heterotrimeric G proteins. As I will show you in the following slides, TREADs are very useful tools for the study of GPCR physiology. TREADs endowed with different coupling properties can be expressed in a cell type specific fashion via different techniques. CNO treatment of these mutant mice then leads to the selective stimulation of distinct GPCR signaling pathways only in the TREAD expressing cells. This approach makes it possible to assess the in vivo consequences of activating specific GPCR signaling pathways in a specific cell type. Shown here are the different metabolically important cell types that we have targeted by TREAD technology in the past. Today, I will present some recent work that deals with the role of GPCR signaling in alpha cells of pancreatic islets. As you all know, pancreatic islets consist of several different cell types. Maybe the most important are the alpha, beta, and delta cells, which make and release glucagon, insulin, and somatostatin respectively. Insulin release from beta cells has been studied in great detail. In contrast, relatively little is known which factors regulate the release of glucagon from pancreatic alpha cells. However, there is considerable evidence that knowing more about this process may be of considerable physiological or clinical relevance. Traditionally, glucagon is considered a hormone that counteracts the physiological functions of insulin, particularly in the liver. Moreover, glucagon plays a key role in the pathophysiologically of type 1 and type 2 diabetes. In type 2 diabetes, elevated plasma glucagon levels contribute to hyperglycemia, particularly under fasting conditions. On the other hand, in type 1 diabetes, impaired glucagon secretion increases the risk and mortality of insulin-induced hypoglycemia. We were interested in exploring whether and to which extent different GPCR signaling pathways modulate glucagon release from pancreatic alpha cells. We initially generated a mouse in which we expressed the GI trait. So the GI trait usually inhibits cellular function, including release of hormone from endocrine cells, selectively in alpha cells of the pancreatic islets. Shown is here the scheme that we used. I don't have the time to go into this in detail. We refer to these mice as a-GID mice for the sake of simplicity. In this slide, we injected control mice as well as a-GID mice with a single dose of CNO at time 0. Strikingly, plasma glucagon levels went down dramatically in the tread mice after stimulating this alpha-GI tread. At the same time, insulin levels were also significantly reduced. Such effects were not seen in the control animals. This slide shows the results of glucose tolerance tests, IP glucose tolerance tests. When injected with glucose alone, the control and the tread mice showed similar blood glucose excursions. However, co-injection of glucose with CNO demonstrated that the alpha-GI tread mice showed pronounced glucose intolerance. During this test, we also measured plasma glucagon and plasma insulin levels. We found that after treatment of mice with glucose plus CNO, again, plasma glucagon levels were drastically reduced, and strikingly, glucose-induced insulin release was severely impaired in the alpha-GI tread mice as compared to the control animals. These data suggest that release of glucagon from pancreatic islets is able to promote glucose-stimulated insulin release effectively. And we also carried out a series of studies with perifused islets prepared from these mutant mice that supported this concept. I don't have the time to show you these data. These data, as well as additional work that we carried out with different receptor blockers, led us to propose the following scheme. The release of glucagon from alpha cells enhances the release of insulin from beta cells, especially when glucose levels are high. This effect is primarily mediated by glucagon stimulation of GLP-1 receptors expressed by beta cells. So it is well known that glucagon cross-reacts with GLP-1 receptors. When glucagon release is inhibited, this pathway is disrupted and insulin release from beta cell is impaired. So these data demonstrate that glucagon has dual opposing actions to maintain glycemia, depending on the nutritional state of the organism. Under fasting conditions, glucagon raises blood glucose levels by promoting hepatic glucose production. In the fat state, glucagon lowers blood glucose levels by enhancing glucose-stimulated insulin secretion by acting on beta cells in a paracrine fashion. We next wanted to explore other G protein pathways that are operative in alpha cells. We next generated a mouse that expressed the GQ trait selectively in pancreatic alpha cells using the mating scheme shown below. In this case, we injected control mice and the alpha GQ trait mice, or short alpha GQD mice, with another trait agonist, DCC. This is a recently developed CNO derivative. It is metabolically more stable than CNO and also is about 100 times more potent. Strikingly, DC treatment of the alpha GQ trait mice represented by the filled bath led to a dramatic increase in plasma glucagon as well as plasma insulin. This was seen both under fed as well as fasting conditions. No such responses were observed in the control mice. Shown next are the results of a glucose tolerance test. When injected with glucose alone, the control and the trait mice showed similar glucose incursions. However, when glucose was co-injected with the new trait agonist, glucose tolerance was significantly improved in the alpha GQD mice. And we also measured plasma glucagon and plasma insulin levels under these conditions. We found that plasma glucagon as well as plasma insulin levels were drastically increased in the trait mice after treatment with glucose and the new trait agonist. And we observed similar results when we carried out studies with perfused isolated islets prepared from control and mutant animals. So these data strongly support the concept that activation of GQ signaling in alpha cells promotes the release of glucagon, which most likely by a paracrine mechanism stimulates the release of insulin from adjacent beta cells. We next wanted to identify endogenous GQ-coupled receptors that are enriched in pancreatic alpha cells of mouse and human. So we analyzed published RNA-seq data, and we identified two receptors, the one highlighted in red, the V1b vasopressin receptor, and the bombesin receptor subtype 3, shown here for the mouse, shown here for human islets, which are highly expressed in alpha cells, but are present at very low levels in beta and delta cells. Shown here are studies where we incubated islets from wild-type mice with a selective V1b receptor agonist or a selective BRS3 receptor agonist. As expected, both agonists strongly stimulated glucagon release. These responses could be blocked in the respective knockout animals. So the purple circles stand for the V1b receptor knockout islets, and the brown circles stand for the BRS3 knockout islets, shown here. Also, not shown here, a selective GQ inhibitor, referred to often as UBO, completely blocked both of these responses, indicating that these effects are truly mediated by GQ type G proteins. We next wanted to explore the potential physiological role of these alpha cell GPCRs. We first focused on the V1b receptor because of its well-characterized pharmacology. It is well known that hypoglycemia leads to an increase in plasma arginine vasopressin levels, also abbreviated as AVP. In agreement with this published work, we showed that insulin injection, which causes hypoglycemia, not shown here, led to a significant increase in plasma AVP. And the same was true when we injected wild-type mice with 2-deoxyglucose, or short 2-DG. This is a glucose anti-metabolite which mimics a state of glucose deficiency or glucopenia. Again, this 2-DG caused a pronounced increase in plasma AVP. In parallel, we also monitored plasma glucagon levels after insulin or 2-DG injection. We found that either compound led to a significant increase in plasma glucagon levels. So shown here. Let me go back one slide. Yeah, so both insulin and 2-DG caused a significant increase in plasma glucagon and, as I mentioned, plasma AVP. So we speculated that this increase in plasma glucagon might be mediated by V1b receptors. And to test this hypothesis, we pre-treated the wild-type mice with either vehicle or a selective V1b receptor agonist, SFR149415, and then injected with insulin or 2-DG. Strikingly, the insulin and 2-DG mediated increases in plasma glucagon, represented by the black circles, were either completely abolished or drastically reduced after pre-treatment with the V1b receptor antagonist. So these data strongly suggest that hypoglycemia leads to an increase in plasma AVP, which then acts on vasopressin 1b receptors, most likely on alpha cell V1b receptors, to promote glucagon release to restore euglycemia. So these findings are summarized here. Activation of GQ-coupled receptors expressed by pancreatic alpha cells strongly stimulates glucagon release. The resulting increase in insulin secretion, most likely caused by paracrine action of glucagon on beta cells, leads to improved glucose homeostasis. Alpha cell V1b vasopressin receptors play an important role in stimulating glucagon release in response to hypoglycemia or glucose deficiency. We speculate that drugs that can target GQ-coupled receptors endogenously expressed by alpha cells may prove useful to regulate glucagon secretion for therapeutic purposes. So this is the last and most important slide that lists the co-workers and collaborators involved in the thread work that we carried out over the past 10 to 15 years. I just want to emphasize that most of the work that I showed today was carried out by a postdoc in the lab, Liu, shown here, and she has reason to smile because the work has been going very well. I hope I was not too fast. Thank you very much for your attention, and I'm happy to take any questions that you may have.